Now, modern development. So I was asked by Professor Tocchini Valentino by Glauco to give you an introduction into the CRISPR case, so to speak, a case which is extremely popular. This is the cover of the most recent Nature of last Friday, where it says CRISPR is the clustered, regularly interspaced short palindromic repeats, about which I will talk a little bit. And the animals up there, from bees to pigs and whatever, elephants, yeah, they all can be manipulated with this modern new tool. Now here, for the occasion, I will divide my talks into three parts. The CRISPR biology, which is basically microbiology, the microbiology with which the system was discovered. And afterwards, only 20 years later, or so almost 25 years later, I'll show you in a second, people realized, Emmanuel Charpentier in particular, realized that that could be developed into a biotechnological tool which is useful for genome editing in almost every organism you can think of. So a very general tool. So how this came about, I will mention tomorrow morning, and why it came about. And then I will talk about, I will also talk tomorrow, as you can see here, about the editing process, but also the recent developments called gene drives, which are quite interesting. And then I will also talk about the moral issue and ethical issues, because this system can be applied not only to pigs and bees and elephants, but also to people, also to us. Human embryos are just as easy to manipulate, probably, as are any other cells, and somatic cells or cells which are used for reproduction. But that will be the last part. Now, the CRISPR development, that's a table which I took from a famous paper from Jennifer Doudna and Charpentier. I come to them a little bit later. You see also they divide such a table, which was published 2 years ago, into CRISPR biology, the upper part here, and to genome editing. It started actually because in a company which produces yogurt and cheese, Danesco, which now belongs to DuPont, but they had observed that when they produce yogurt, that occasionally they lost all their cultures due to phage infection, bacteriophage infection. Now that's a serious economic problem, as you can imagine. If you lose 50,000 cubic meter, 50 million liters of yogurt, this is quite expensive. And so they started to investigate the problem behind this phage infection. Why does it happen that these cultures get destroyed? And if you look here on this table, you can see that the CRISPR system itself was discovered in '87, and then it was discovered that it contains a sequence of important genes, relevant genes for bacterial immunity. In fact, it turns out that this is a mechanism of innate immunity of bacteria. And then it was discovered that there are several types of these immunity mechanisms. One is the type 2, which was identified as being the most simplest type, which could be adapted then to use as a biotechnological tool. And at the same time, genome editing was developed, and some of you here who are not, who are younger than me, but or even my age, they realized that there were lots of ways of how to edit genomes. That was always of interest, to modify the genes in any organisms, in yeast or in mice. And that started with very difficult analysis and in yeast, and then zinc fingers and so on. I will come to that later. And then everything came together around 2011 when it was shown that small RNAs, and this is the course on RNA biology, small RNAs are responsible for this immunity process. And ever since has become incredibly popular. It's a craze. Everybody talks about it. And this is a table from PubMed. You can see that the red is the exponential curve. And you can see that the number of publications follows the exponential curve. In fact, in 15, which is not on here, I looked it up. It's more than doubling of the publications. So it's about 1,500 publications last year which contained the title or in the abstracts CRISPR-CAS. So it looks as if everybody's using it. Now, let's turn to the biology before we come to the CRISPR craze and go back to the beginnings. In the beginnings, the problem, as I said, was a problem of phage resistance or phage sensitivity. Cultures which are economically important for cheese production or yogurt production, like Streptococcus thermophilus or Biogenesis. I always, when I talk to the general public about these things, they are always shocked to know that yogurt is nothing else than a slur of bacteria. They don't really want to know this, but it is anyway. And so the matter was studied for economic reasons here. That was the start, yogurt, cheese, other fermentations, and then for health reasons, because there was a time, and there may be coming a time, when it was thought that you could treat bacterial infections using phage infections and kill the bacteria through bacteriophage. That is completely out of fashion these days, but it's maybe coming back because there's so much problems with antibiotic resistance, as you know, that but there are very few people in the world. There are only 2 labs in the world which kept all along all these different strains of bacteriophage. There's a lady in Tbilisi in Georgia who has a collection of 1,600 of these phage strains, and she's becoming popular, I think, now, because nobody has anything left otherwise. And even glycosylphages, which we grew up, like T4 or Lambda or T7, are very difficult to come by these days. Everybody cleaned their refrigerators. So health reasons, and then of course it's the microbiologists want to keep original bacterial lineages because they want to have phage-insensitive hosts in order to keep their collections alive. That's also an interesting point. So how do phage infect bacteria? That's a replication cycle, and the phage absorb, as you hope. They absorb here on the surface of the bacteria, DNA entry, replication, and so on, transcription, assembly, and the host cell dies. There, of course, there's a replication cycle, which is not really a replication cycle, where the phage becomes lysogenic, that is, the genome becomes integrated into bacteria, but I will not discuss that here. Now, the bacteria have a number of mechanisms to defend themselves against phage infection. And the one we are talking about here is after DNA entry, the DNA of the bacteria of the phage is inside the bacteria. So, and there is, of course, a very old mechanism, restriction modification, which was discovered in the 50s, Bertani and others in Stockholm, and that led to the first phase of recombinant DNA research, restriction enzymes, which of course are enzymes which destroy DNA, and now you can buy them everywhere. But I'm not going to talk about this restriction modification system. But the other system is new, and the other system is the so-called CRISPR system, which I will now get into. So what happens schematically is that the phage infect, this is the surface of a bacteria, infect their DNA into the inside of a bacteria, and there are certain sequences, which I come to in a minute, which are between 30 and 40 nucleotides long, so-called protospacer sequences, which are integrated somehow, I will come to that later, which are integrated into the CRISPR site, and I come to that again, so that the bacterial genome now contains this little piece of the bacteria of the viral DNA. You know, this piece ends suddenly up in the genome of the bacteria, and then in the next step, and we will see this how it works with nuclease and so on. Because this piece is present here, it can destroy any DNA, any phage DNA, incoming DNA, which contains the same sequence, the same spacer sequences, which has been taken up here. And then if there's no protospacer sequences, then the phage can infect and kill the bacteria. Now these CRISPR loci, as they are called, these clusters of repeats, I think I showed at the beginning the exact abbreviation, but I will only talk here now about CRISPR. These repeats are, this is the first one which was ever discovered. That was in Streptococcus thermophilus, an old paper, 2005, but it shows the general principle. To the left here are all these CRISPR-associated genes and proteins, which have different numbers. I will come to that again. And these are the CRISPR-associated compounds are mainly nucleases, because they are later on needed to destroy the incoming DNA. Then there is this CRISPR region, which looks very funny here. This is a duplication of repeats and the spacer in between. Everything around between 30 and 50 nucleotides, which are clusters, which are repeat 30, 40, 50 times. This is why it looks so funny here. And then there's other genes which are not related to the problem. And usually bacteria have two clusters of these peculiar, one or two, not more, of these peculiar elements here. That is the CRISPR element, this cluster of repeats, which, as you will see in a minute, these repeats are actually partially palindromic. This is just a few examples here. You don't have to memorize them, but you can see they're everywhere in the bacterial kingdom, even in Archae bacteria, and they can form these hairpins because they are palindromic. This is why the name is, the S and the P here is short palindromic repeats. They are usually, they are not really very big, but they are indispensable for the process. If there are no repeats, palindromes, then the thing doesn't work, because we'll see later how this happens. But this is not the only structural element which is necessary. By the way, the sequence, as you can see from these logos here, the sequences are diverse, but they are G-rich and so on, but they have to be palindromic. But there's one other element, and adjacent to this, you see here it's 32 nucleotides, sometimes it's 35, sometimes 31. Half of all bacteria contain these sequences. It's quite variable, but in that order of magnitude. But there's another element. At the end of each of these repeats here is 31 here, and is an element called PAM. protospacer adjacent motif. You will see in a minute. Protospacer is that region which is adopted from the incoming phage, this 35 or so nucleotide, 32. And adjacent is always a sequence which is kind of NGG, AGG here, and that sequence, you see the logos here, they are not always fully conserved, but they are of this kind of general structures, 3 nucleotides. And if there's no PAM, if there's no protospacer adjacent motif, then things don't work. So you always, that becomes important tomorrow when we talk about the genome editing, these guide RNAs, the RNAs which guide the nucleases to the proper regions of the genome, they require always this kind of elements. Otherwise, it simply doesn't work. So that is important. But of course, it's crucial, indispensable for a bacterial infection or for a phage infection to make it resistant. And actually, the PAM sequence, the protospacer adjacent motif, determines the spacer orientation. You see, it's always in front of the protospacer and not in the back. So if it's here, it looks as if it's in the back, so then it turns around, so that it's always transcribed in this direction. So it's a real crucial element, these 3 nucleotides. Never forget PAMS if you ever do a CRISPR experiment. Of course, if you buy the kits today, the problem is you can buy everything now. I looked at it on this morning, the latest catalog of genes, the simplest kit is 180 euros you can buy. So it's probably most people who use it don't even know what they're doing, but that's another problem. That's true with other things. You know, I'm now off limits, so to speak. But I wanted to come back after 20 years of running funding agency. I want to come back to do experiments myself. So I wanted to practice a little bit. I wanted to isolate the DNA from yeast. And so I started, and a group leader from an ERC starting grant here in Italian, Fabiana Perocchi, working on mitochondria. She gave me a graduate student to help me. I said I can do it anyhow, but she helped me. So I started to get to ask her, get me some sodium acetate and please make a 1 molar solution of sodium acetate. She didn't know what to do. She didn't know what to do. I said, well, you look up the molecular weight and then use a balance and weigh. We don't have a balance in the office, in the lab, no balance. I hope you have some balances here. And then I needed a 10% SDS solution, which I could only get because in some kits there was a 10% SDS solution. If not, she would not have known what to do. So the problem with the kits is really serious. Anyway, the PAM, the protospacer, is always important in order to make this bit. And this is the entire array. You see here in gray, the protospacer, which is the sequence which comes from the phage, and then the sequence, the repeats in black here, which I've shown you, have to be palindromic, at least in part have to be palindromic. Now, and then you can see what happens. This is how CRISPR immunity is happening. Here is a phage injecting its DNA into the cell. I took that from a recent review. Barangou, by the way, is the one who, he's now a professor at NYU, but he used to be the head of research at Danesco, this company who was in trouble losing all their yogurt production. So there's a very good review. Actually, I mentioned always where these figures comes from, so that you can look them up. And so what happens is, so the new spacer is coming in here, and you see it here now. in green, and what you see already is the crucial business here is that this operon here, as you may call it, is transcribed into RNA, in the so-called CRISPR-RNA here. The CRISPR-RNA, as we will see, is not just this short piece of RNA, which is 3530. Oh, it says here. It's 20. Yeah, here you see the size of these repeats. But it's made as a precursor. We will come to that in a second. And then it's cut down by a nuclease into these small pieces, and these pieces bind to a nuclease here in blue, and this then cuts the phage DNA, and then the phage cannot infect. It's really very simple. But it's a mechanism which requires a lot of RNA, which requires RNA. And here you see again the protospacer and the PAMS sequence, which I mentioned. So that is a more general view of the system. 45% of bacteria and 83% of Archae have this sequence, and therefore have these mechanisms of adaptive immunity. It's actually a real adaptive immunity mechanism, very efficient. And there's a lot of variation in these systems. Here, this figure shows two variations. Is that actually focused? Can you see anything? Is it focused, or should we focus? Anyway, you see, here you see for the first time, you have the CRISPR array with these protospacers and the repeats in blue here, in light blue. And this is made into RNA, into a long precursor, and that is processed. And the mechanism of how this is processed is very different in different systems. We come to that in a second. And in the end, in red are marked the sites where the phage DNA then is destroyed with the red arrows here. You can already see here that there's a special case here, Streptococcus pyogenes. Because that is the case, a so-called type 2 case, I'll come to that in a minute, which is later on used in these genome editing procedures. So there are many types. There are several types, and it's already indicated here there's a new type of RNA here, which is being used, and that is a very important, that is actually crucial to all this process, which we are going to talk about. So we have actually three types of mechanisms. In all cases, there is a pre-CRISPR RNA, which is RNase polymerase 3 produced a long RNA from these funny CRISPR sequences. You see the green and the red and the blue are all these protospacer sequences, and there can be 30, 40, 50 in a row of those. And they are processed into smaller. First, in type 1, there's an enzyme complex in light green here, which has at least 7 enzymes. It's a rather complex, so-called cascade it's called. And 2 of these are well characterized. The first one, CRISPR-associated genes or proteins. They cut the long RNA precursor. And then comes another enzyme, which in connection with PAM binds to the sequence, to the protospacer sequence, and cuts. So in this case, it's at least 2 enzymes in type 2, in type 1. The most interesting one is type 2 here, where again, but I come to that in a minute. There is not just one RNA, one precursor, CRISPR, CR is for CRISPR, but there's a second one which shows suddenly up, TRAC-RNA, which I will just explain. And then the 2 of them get together and then cut the DNA. This is the type 2 system, and the type 3 system is rather similar to the type 1 system in a very general term, but I will not discuss it here in detail. Now, the crucial is the type 2, and the discovery in 2010 of not just the production of this CRISPR-RNA, that is an RNA copy of these peculiar repeats, which I've talked about, but there's another RNA which is crucial. Where does this come from? Now that was I will describe how this works, how that was discovered even, because I consider this the crucial paper and the crucial piece of work. Was done by was done by Charpentier and some other people, which we'll see in a minute. And the experiment was, or the hypothesis was, maybe there is another RNA, and how could we identify it in the mountains of RNA which are produced in bacterial genomes? And there was somebody in Würzburg in Germany, Vogel, who had discovered a way of analyzing this using so-called differential RNA sequencing. So what they did was what they did was, this is just an example from the catalog of the company from whom you buy Terminator exonuclease. It's an exonuclease which digests RNA as long as it doesn't have a 5' triphosphate. All the other breakdown products and everything which you find in an interior of a bacterial cell is broken down. That is, you enrich in RNA which has a proper start site, the 5'. And so here you see all the large ribosomal RNA, for example, disappears. After treated, it's all gone. So you have enriched your extract in messenger RNA in this particular case. Now, in the case of the small RNAs we are talking about, there's the same problem. They also start properly with a 5' triphosphate, but there's of course loss of background, so you have to get rid of this too. The first paper for this interesting technology, and I can only, I think, I don't know whether you learn this here in the course now, but it is really enriching by a factor of 50 to 100, 2 orders of magnitude almost. Without it, you couldn't even see anything. It was done in a paper, it was first described in a paper on Helicobacter pylori, the bacteria which are in the stomach responsible for ulcers. You can see here very well no exonuclease in black with exonuclease, and you can see how in the presence of exonuclease the start sites are highly enriched. Here the red positions here, and there are some other ones here. This has nothing to do with CRISPR now. This is just a way to determine the transcriptional start sites in the genome of Helicobacter. There are actually 1,010 have been discovered in this particular paper, which is mentioned here somewhere. But it's the best example of how to enrich in 5' PPP phosphates and RNAs. And that technology was, in a very famous paper, was applied to the CRISPR problem. And then you can see here in BIOGENESIS, you can see here that this is what we have seen before. This is the CRISPR repeat, which is made into RNA. First in a precursor, I think I can read here 511, and then it's cut down to 39 and 42. CRISPR RNA, a copy from these repeats. And there to the left is all these nucleases. And in the case of type 2, it's this one is, it actually was called at that time CSN1, but it's now called Cas9 in all the literature, but I come to that. But then they discovered there is a transcript to the left. This is transcribed to the right, as you can see here, the arrows. And this is transcribed to the left, and it's called the TRAC RNA, because the RNA is necessary for the maturation of CRISPR RNA. So this is not just simply cut, but it has to be, there has to be a complex of this nuclease together with this RNA which is transcribed to the left. So the secret of the CRISPR-Cas9 system is that you need two RNAs, two small ones. The RNA which contains these protospacers and the repeats, and the RNA which is transcribed from here. Turns out that they only they hybridize together. They have a certain homology, as you can see here by the black sequences, which was discovered in this process. And only if they base pair can the DNA later be cut into pieces. And here is just the experiments which show that this is processed from a bigger precursor to smaller precursors. So the type 2 system, to come back, requires two RNAs, two small RNAs, not just one, but two. And they are dependent from each other. So they are made, as I said here before, both RNAs, this is the CRISPR RNA, which has which is this long protospacer, spacer, and so on. And in order to digest it into small pieces, you need the other RNA. The other RNA, which is transcribed to the left, you remember it, is required, binds to the enzyme. In this case, it's called, in the paper, it's called CSN1, but it's actually Cas9. In 2012 or so, they changed the nomenclature, and therefore everybody talks of Cas9. And then this complex binds each other. You see here is the long precursor, green, black, black, green, black, and the red one, the TRAC RNA, binds to that by hybridization together. And I showed you that they have a short stretch of sequence homology, and then they bind to each other, and then the DNA processing enzyme, the RNA processing enzyme, binds and cuts it eventually to 39 to 42 nucleotides. So in order to cut the RNA, you need you need another RNA, and just without this, it doesn't work. And that is crucial now for the development of the genome editing tool. Now this arrangement in type 2 is coming in many bacteria. You see here to the left is this TRAC DNA BIOGENESIS. This is the famous model case, but there are others, even highly pathogenic strains. And they always have this TRAC RNA, which is transcribed to the left and which is necessary to process the RNA which is transcribed from the right side. And they're sometimes of different size, as you can see, but in principle the arrangement is very different. There's always Cas9, CSN1 in the old nomenclature, but there's always Cas9 here. And the others are not used in this particular process. It's often not clear why there are why there's more than one in the type 2 systems. In the others, it's much clearer because you need several of the Cas enzymes to do the job. Here, you need only one, but you need this extra RNA. That's the big difference. So in all the other cases, type 1 and type 2, you need this cascade complex, which has many nucleases. And it's only in this one you need one nuclease, but that is guided by two RNAs, the one in red and the one which is transcribed to the right here. Now, in order to look at this in a schematic way, so that was a crucial paper which discovered this, that in type 2 you have these two RNAs to do the job. Then she got together with, then they had a joint postdoc, Martin Winnacker, who's now an assistant professor in Zurich now. But at that time, they got together, and here's the system. You have the two RNAs. The CRISPR RNA, the short piece, processed from a long precursor down to this 35 or so nucleotides. And you have the other RNA, here again in red, the so-called TRAC RNA. And they have some homology here. 15 nucleotides, 16, so varies a little bit. And you can see, so you have the system actually is a 3-component system. The nuclease, here in blue, which has different domains, we come to that too, and the 2 RNAs. They have, these 3 components have to come together to make it into to cut the DNA, to cut the incoming DNA, and here is where it is cut. And you see always this protospacer and the protospacer adjacent motif in red here. That, if that isn't there, you cannot do anything. But of course now, as I described to you, it is well known now. And then the 2 of them somehow got the idea, why not link the 2 here? See, here they are separate. But you could, they had together the idea, I think Charpentier told me during a meeting in Cold Spring Harbor, they made a walk on the beach and had the idea, why not link them together? So that you have a single RNA which contains all the necessary elements, except instead of 3, you have only 2 elements now. They are linked together here. By a short linker, it's only 6 nucleotides or so. And so instead of 3 components, you have only 2 components. And that makes all the difference. And this is this is it. This is CRISPR-Cas9. And that made it possible to use it in every potential system which you can think of. Now the nuclease, this Cas9 is an interesting, it has by now it's been crystallized, as you can imagine. And this is just a schematic view because the crystal structure, you can just look it up here. But I didn't have this possibility to make this into a movie. You need a special programs to do that, which I thought I wouldn't work here, so I didn't bring it. But you can just look at it at that website. So you can see this Cas9 has a lot of subunits, which are known from other systems. And it has that H and H motif, it has the RAF-C motif as the CTD. That is the sequence where the RNA polymerase binds. Come to that tomorrow, I think. And then with the RNA binding, you get huge conformational changes, as is indicated here. Kind of comes together, the 2 things. And it is now known where the PAM recognition sequence is, and how the RNA, how the 2 RNAs recognized together with Cas the DNA and cut it into pieces, and the amino acids are already known. Here is this peculiar PAM sequence with the 2 Gs. I mentioned in the beginning of my presentation, you need this NGG sequence to do, you always need it. And yeah. So a complex, actually the Cas has, the Cas9, which almost everybody works with, has 1,031 amino acids. So it's quite a rather big protein. This one, which has been crystallized. Now the race is for smaller ones, which are smaller than 1,000 amino acids, because you want to fit them, as I will show tomorrow, into, for example, adeno-associated virus, which is a virus, which is like a ball, but there's not much space in it. So you better need a smaller nuclease. And there's one now has been described which has only 242, I think, amino acids. They are just looking every bacterial strains now. 1,000 against. The small one is Zhang Feng at the Broad Institute. There's a big, well, I haven't come to that yet. There's a huge patent discussion going on. In fact, the US Patent Office has started an interference process, as they call it, because well, it's not very scientific now, but this is just clutch. But it turns out that Doudna and Charpentier patented first, and then came the Broad Institute in Cambridge, Massachusetts, which is a very famous place, where Zheng Fang is and others, and they were later. But in America, since 2012, you can speed up the process of getting a patent with money. So if you put 250K on the table, they take your patent from the bottom of the pile to the top of the pile, and then they got the patent already half a year ago. And the others are still waiting, even though they filed much earlier. And that is now subject to an interference procedure. I mean, in court, it's like on the high seas, as they say, you never can predict the outcome. And of course, there's hundreds of millions of dollars involved. But that's not my subject here tonight. So that is what I wanted to tell you about the biology. These are the three important papers, I think, which describe everything, which are the clues. The first paper, which describes, which I told you, why bacteria become resistant to certain bacteriophage, because of this acquisition of these short sequences, the so-called protospacers, from the bacterial genome from the phage genome onto the bacterial genome. Now the biology of this process, how these pieces of DNA get in there, is not known yet. People have not yet worked out this. So there's a lot of work to do. But it works, and that was found out by Rodolphe Barangou in 2007, still at Danesco, this company which you're all eating yogurt all the time, even today. The second paper is the crucial paper with Charpentier, where they found this TRAC-RNA, the second RNA which you need to get this whole process going, which is only used in type 2 systems, but makes it so simple. And they use this differential RNA treatment, which I tried to describe, where you had three PPP, 3 triphosphate ends. And then, of course, the paper in 2012, where they got together, Doudna and Charpentier, to develop this tool whereby the 2 RNAs are fused together. I think I tried to describe this. So that a 3-component system becomes only a 2-component system. And these are the bacteria which we are working with, just so that you know what we are talking about here. So this is yogurt at its best here. I don't know why the photo is green, but I took it from some friend. No, Emmanuel sent it to me, actually, probably the GFP. And these are the photos of these people. Charpentier, here on the left. She's now in Berlin, yeah. Max Planck, Doudna in Berkeley. And this is Rodolphe Barangou, now at, not NYU, but North Carolina, sorry. North Carolina University, and before that in Damascus. And this is this Whiskey Church whom I come to tomorrow, who wants now to clone a mammoth. And I'll explain how to do that. Good experiment for ICGEB, maybe. And of course, the New Yorker already made a story out of this. I don't have to explain this in more detail. I think that's what I wanted to say today. And tomorrow we then come into the actual applications of the system, and later of the moral issues. So now you know the biology and the biological background of the system. Thank you.
Thank you very much.